kinase. Now protein kinase. So as we can see, protein kinase, there are several type of protein kinase present in our cell, like tyrosine protein kinase, serine protein kinase, threonine protein kinase. So suppose here we take an example, and so that we can understand it better. So for example, the SRC gene product is a non-receptor protein kinase called PP60 SRC protein kinase add phosphate group to the amino acid tyrosine. Protein phosphorylation was known to be important in effecting a multitude of metabolic changes in the cell. SRC and other tyrosine protein kinase coding oncogenes might transform a normal cell into a metabolically different cancer cell. So now we understand that SRC is a we take an example of SRC gene, that is a proto-oncogene, and it is a protein kinase. It is a non-receptor protein kinase. So SRC gene encode a protein that is PP60 SRC protein. This is not important. The name of the protein is not important because there are two types, PP60 VSRC, this is viral SRC, viral oncogene in case of viral, and there is present PP60 CSRC in case of cellular SRC. So this is viral SRC, this is cellular SRC. This is not important that much, that name of the protein, but you have to remember that the name of the gene tells is SRC. So SRC protein kinase add phosphate group to the amino acid tyrosine. So it phosphorylate on a tyrosine residue. So this is a protein. So it phosphorylate on tyrosine residue, specific tyrosine. So a tyrosine residue. Now protein was known to be important. When SRC encode a protein kinase, and the name of this protein is PP60 SRC protein, and it phosphorylates some protein that affecting a multitude of metabolic changes in the cell. So the different changes metabolic changes occur within the cell. Changes occur within the cell. So SRC and other tyrosine protein kinase coding oncogenes might transform a normal cell into a metabolically different cancer cell. So SRC and other tyrosine protein kinase coding oncogene. Suppose there is a mutation in the SRC gene, and it leads to excessive phosphorylations of that particular. So if there is a mutation in this particular SRC gene, and now SRC gene acts as an oncogene, and this oncogene helps in the phosphorylations of different protein, and now the normal cell loses its function, and metabolically different a multitude of metabolic changes in the cells occurred, and that ultimately transform a normal cell into a cancer cell. So if there, in case of normal, SRC encode a protein that helps in the phosphorylations of a protein in specific tyrosine residue, and there is metabolic changes occur within the cell. There is some metabolic expressions of something, or some genes occur, but if there is a mutation in the SRC gene, the expressions of the SRC gene increases, and it may leads to phosphorylations. It constitutively phosphorylates a different type of protein due to phosphorylations of different type of protein, it leads to change a normal cell into a cancer cell, and ultimately a cancer is formed. So this is the whole process, like growth factor, protein kinase, and earlier we discussed about the membrane associated proteins. These are examples of the proto-oncogenes that when mutations occur in such type of proto-oncogenes like membrane associated G protein, RAS, in case of growth factor, like we take an example of PDF. In case of protein kinase, we take a SRC gene. So mutation in this type of proto-oncogene leads to transform a oncogene, and when a normal cell bears oncogene, it loses its normal function and transform a cancer cell. Thank you. If you like the video, please share.